Hello everyone. So today I'm going to be guiding you through how to perform an analysis of a 2D-DIGE experiment within our SameSpots software. Now, the vast majority of people that are doing 2D page, 2D gel-based experiments these days are using 2D-DIGE or 2D-DIVE. They do this to minimize gel-to-gel variation, which is a large source of variation within the technique. And if you can perform, 2D-DIGE using internal standards, you can remove that gel-to-gel variation as much as possible to ensure that you've got no missing values. Again, SameSpots excels in producing no missing values whatsoever. But it also reduces inter-user and inter-gel variability, so your experiments are much more reproducible, much more rigorous. So your data is much more reproducible and much more rigorous. SameSpots only comes in one version. Now, other software in this space try to upsell you if you want to do DIGE work and will only support single stain in the base version, and you kind of have modules that you add on and things like that. We don't do that. Our SameSpots software is designed for all 2D proteomic analysis, 2D differential expression analysis. So we do support straight out of the box with one license, single stain, multiple stains, DIGE, and the various spot check and gold standards that we've got down here that I'll probably explore in another video, but they are designed to make sure that your experiments are really reproducible, really rigorous, and designed for very large data sets, complete population proteomic studies and very large data type studies. So I'm going to come straight in. This is the loading screen. This is the first screen you get when you go into SameSpots, saying, do you want to, what files do you want to open, what experiment do you want to do today? If I have a 2D-DIGE experiment with internal standards, which I do have a set of images today, I'm going to click on DIGE, create my new experiment. So let's just call this 2D-DIGE YouTube demo. I'm not going to copy spots from an archived experiment. Again, this is a feature whereby you can copy spots from other experiments to see how they compare to your current experiment. Again, part of the toolkit that we have to support very large population-wide studies with hundreds of thousands of samples and be able to copy them between experiments and see how things have changed in very large data sets. So I'll create the experiment. And the first thing we need to do is add some images. So I have a set of images that I can use for this. Here we go. So you can see I've got, well, you won't be able to see this because I'm going to blur it out so you can't see all my files. Once I import these images, you will be able to see it though. So the first thing SameSpots does is it examines all of your images to make sure they're of reasonable quality. The reason we do this is poor quality images are going to give you poor quality results, and we want to tell you straight away before you get downstream, before you get your data in front of you, if these images are of a high enough quality to produce high enough quality results. So you don't waste your time as an operator analyzing poor data and making assumptions, rejecting, accepting hypotheses, things like that, based on bad data. So you can see it says no problems found here. So within this data set, all of the images are, 16-bit. The dynamic range is in use but not saturated. The image is in grayscale. It's in a TIFF format, so it's not been compressed. It's not got any compression artifacts or false values put in here. If I did want to perform any rotations or any crops or any image editing at this point, I can do with these tools down here, but I don't need to for any of these images. So I'm going to go ahead and just include all of these by ticking them. I can actually probably just select them all at once. My laptop's going a little bit slow here. Probably because I'm trying to open all these images at once. I'll just turn all of these on to say that I want to include them within my experiment. Now, if I did have any failures, I could exclude them at this point. So I can keep my images in the project file, but having chose to exclude them. So you can see that someone has made a choice based on the data that they've put in. They've not just missed an image file to put in. A judgment call has been made and there's evidence of that. So you can see here my images, gel 1 control, drug A, internal standard, control, drug A, internal standard. So the setup I've got here is 4 gels. My CY3 channel is my control non-treated data set. My drug A CY5 labeled set is, a group of people, animals, whatever it may be, cells treated with drug A, and my IS-CUI2 labeled IS is my internal standard. So what happens is next I will go through the process of telling the software how my experiment is configured. So how have I orchestrated my experiment? So I'm going to add gel 1, gel 2, gel 3, and just select all of these and then add them to the gel that this lines up with. So I'm just holding control here so I can do a multi-selection. Add those to gel 1, gel 2, and this just tells the software basically what to compare to what. It'll make more sense as we go further down the line. And I need a gel 4 here. So what will happen now is the software knows these are meant to be the same gel and they have an internal standard. And I can mark my internal standard by clicking this button here. So what the software has actually done is it's automatically, because I've named it with IS in the center, it's automatically picked out and labeled my internal standards for these gels. So it already knows that what we want to do is compare drug B and drug C for gel 4 to the internal standard for gel 4. So it's already jumped ahead and kind of worked out what you want to be able to extrapolate from this experiment, what we want to know. And what it will do is as well, when we're choosing this reference image here, is this is for the automatic alignment that's built into the software. What the software will do is align all of your gel images to your internal standard. And to the internal standard, you can choose which internal standard you want to align it to, but the software will automatically scan all of your images and detect which it thinks is the best internal standard to use as the template for the automatic alignment. So already a lot of these decisions are made for you. You can, turn this off. You can override it and say, no, I want to use this one. But I can see certainly for this one, we can see, you might not be able to see it on the video, but there's a thumbprint on the gel here. So that's probably why that's been discounted. But the software makes a very clever judgment based on the intensity of spots, how many spots are on a gel, things like that, where the edges of the gel lie and how crisp they are, that all factor into how well it thinks it can perform an automatic alignment, what is going to give you the best chances of the best quality alignment. So again, completely automated. Next step. Now we can mask off any areas of the gel that we don't want to consider. So if we had any rough edges, if we had any tears, if we had any areas of the gel that we don't want to include either in our analysis or in our automatic alignment algorithm. For example, if you had some writing down the bottom here, which was very intense, the software may pick that up as a spot and it may try and analyze that, and it may use that information to influence how the automatic alignment is performed, which you don't want because you want your alignment to be based on spots and data rather than some black sharpie in the bottom corner of your image. So what you can do is you can just mask off the edges. It's the default tool by drawing a little square here. It's important to note that everything in pink is going to be excluded, and everything that is not pink is the area to be included on this tool. So edges is designed to only exclude the edges. The next step we come through is alignment. Now, of course, we can use manual alignment if we wish to, but I'm not going to go through that within this video. I think it would just make this video too long. I'm just going to focus on automatic alignment, but I may do another video on manual alignment vectors, how you qualify your alignment, things like that at another point. So what I'm just going to do is press automatic alignment, and the software will tell me, hey, look, I'm going to align all these things. It's probably going to take me a second because my laptop is not the world's fastest. But if you were doing this kind of work routinely, you'd obviously have kind of more powerful Proteomics PC that would just absolutely fly through these images. So you can see here, all of these blue spots are vectors for alignment that the software has extrapolated and overlaid on top of each other. Now, automatic alignment works really well with DIGE images because essentially the proteins are very The protein spots are very close to each other already because they're all immobilized in the same gel. So it's very accurate to use automatic alignment here, but of course, at TotalLab, all of our software, we try and automate the process as much as possible, but we don't stop users from having free will and free reign over their experiments, so you can add manual vectors if you feel you want to. But you can see here, in the space of about 30 seconds, it's added near enough 800 to 900 vectors per image. With each vector being done manually, that would take many hours of very tedious work to reach those kind of numbers of alignment vectors. So the next step we'll come to is detecting spots and filtering out spots that we're interested in. So I'm going to choose to detect spots on all of my images. And again, automatic detection, it's going to run through, detect all of our spots. We can then manually add or remove spots based on the spots that are detected. So again, we try and automate as much of the process as possible. The value of this is obviously saving time and reducing operator bias on the data, but we don't stop anyone from adding their own spots or removing spots as well as part of the whole toolkit that we offer. So you can see here, we've pulled out, all of our images from all of our spots from our first image. We can view all of our images separately here and see how the spots match up with each other. And what we can also do from this point on is perform our filtering. It's worth it to note the ones around the edge here have been masked off using the mask that I used before, so that's why they weren't detected. But we have got the edge of the gel here being detected. Now, what I can do is just kind of select these and delete them immediately because I know I don't want to consider them, or I can filter and mass delete things that are kind of weak signals or signals that we know we're not going to consider. So, for example, this spot here doesn't actually seem to have anything in it. So what I can do is I can right-click this spot here and actually use its volume as a filter. So now I can turn on this box and say, right, I want to filter out all of the spots that have a normalized volume less than this spot that I clicked here. So from here, I just turn off the selected area. So you can see here, it's highlighted all the spots that have an equal to or less than spot volume than the one I identified. So I can delete those straight away. If my SOP, if my protocol, if my experience tells me that anything below that is not really a positive result, I can remove those straight away. So again, we've already saved a huge amount of time of having to manually review every spot and go through lots of false positives, lots of spots with zero volume. So next I'm going to tell the software how I organized my experiment. So it's pretty self-explanatory. So do we have samples? Do we have samples from only one group? So we've got drug A, drug B, drug B, drug C, drug D, things like that. Have I got the same samples under different conditions? And then do I have samples that from the same subject that appear in two different conditions? And all this changes is how we set up the experiment and what kind of statistical tests are applied to the numbers that we get from that experiment. I think I'm just going to keep it between subject now. So what the software will ask me to do now is to add the gels to the different conditions. So I can have my conditions as control. I can have drug A. I know some aren't. But what I can start to do is add them to the condition that they're meant to go in. So got drug A, gel 2 control, right, we'll put that in the control group. Got drug A here. And then if I had set up, we could probably add, yeah, there we go. Drug B, so this can go into drug B. And I can start considering these samples as different groups, different treatment groups. Oh no, sorry, that was drug C. Drug B there. So you can see I'm starting to build up a picture of what I want my answer to be, or what I'm trying to discover. So an easy one would be, how are the spots different? How are certain proteins differentially expressed in drug C, drug B, drug A versus control? How does that change things? So the next step I'll come through to is reviewing all of my different spots. So you can see the software and you can change the size of these images up here just to get a better view. If you've got a large monitor, that would be great for being able to review this. So you can see all of your gels in all of your control groups side by side. So you can start to piece together your experiments. And at the bottom here, we've got the expression profile. So in control and drug group B, there's very little change, but actually in drug A and drug C treatment, you can see that the expression of whatever protein this spot aligns with has increased. I don't know if it's significant or not. Probably is. Confidence variable. Yes. So there's a 2.8-fold change, and it's given me p-value of what? Very much below kind of 0.05. So it's below, I can't read scientific notation these days, but it's 0.000002. Well within, statistically significant bounds. What we can do from here is we can filter based on the expression levels and whether it's significant change or not using these different filters. So, using our quick tags. So if I right click, if I select all of my results, right click, go my quick tags, I can say, right, any spot that has a p-value less than 0.05, in fact I might go 0.001 because there's lots on this. I can create a tag and tag those spots. Then I can also layer that up with another tag. So I can say, right, well, I'm interested only in significant changes in protein expression, and I want that change to be at least a 2-fold change. From there, I can then create filters and show all of those spots. And I can use filters to hide spots as well, to be able to shrink this list down to only the spots that I consider to be worthwhile, interesting to look at. Maybe I want to do it in gel digest, do some mass spec and identify these proteins. But certainly I don't have to look at all, 1,000, 2,000 spots and kind of work my way through all of them hoping to find a good result. The software will actually reduce this list right down for you. So we can see here we've got a range of significant changes in protein expression across all of our groups. And that fold change goes from, an 11-fold change in expression all the way down to exactly 2. So this is almost undetectable with the human eye, but obviously the software has been able to extrapolate a significant difference based on the pixel densities at these points, pixel intensities at these points. So from this point onwards, we can start do, principal component analysis. We can do dendrogram statistical analysis to see correlations between changes in expression of different proteins and how they relate to each other. So this might give us some explanation as to different mechanisms that are involved with upregulation or downregulation of proteins when treated with certain drugs. And we can also do power analysis to ascertain whether we have a large enough sample size to be able to accurately make any claims based on the evidence that we've been given. So do we have a large enough population to rule out the chance of variation or error within our results? So that's all calculated automatically based on the decisions that you made earlier. You can kind of select all your different spots here or select all your different areas here and have a look at your standardized expression profiles for all of your spots. You've got access to be able to add all of these images to your clip gallery. So you can export them to papers, put them on posters, you can put them in presentations, just as you can with all of our other software. You can access the ANOVA table, so you can, again, this is a pretty standard format. You can then export this straight into your paper by adding it to the clip gallery to show exactly how, if you're producing a paper and you're making claims about your results, this is exactly how you got there. These are your methods. We do include access, we do, if you do want to do an in-gel digest and you do have access to either manual picking or automated spot picking systems, we do support integration with those. But I'm not going to go through that right now, but we do support that as well. And then finally, as part of our report, we can turn on or off all of the different spots that we've kind of whittled down our list to. We can add spot details, we can add the table, molecular weight, if we've used, if we've calibrated it with ladders or isoelectric point markers. We can include molecular weight markers, protein description, protein accession numbers if they are known. So what you can do is if you have certain template standards or you know where some of your proteins are, you know what they are, you can feed that into the software to identify those proteins and have them be labeled. We can include notes, we can include experimental design, so we can show how we've compared different groups, different treatment groups, different groups of gels, that kind of thing. And we can also add any image montages we want alongside that. And from here we can create a report. So I'm just going to include everything. And then once that report has been generated, you'll get access to that. So it's generated in a HTML format, but this can of course be printed onto paper or printed to a PDF, and you get all of your results, all of your images, all of your expression profiles that I showed you before. So you've got a full summary of how your proteins have changed, how your expression has changed in your different treatment groups for your proteomic work, to then be able to build up your experiment, make your claims in your papers, report to seniors if you're working in like a high throughput proteomics lab. So this is just a whistle-stop tour of how SameSpots can be used in 2D-DIGE techniques without any kind of add-ons or upsells as part of your license. This is built straight into the software. But yeah, that's how we handle image analysis or 2D-DIGE analysis within our SameSpots software in the field of 2D gel-based proteomics. As ever, thank you very much for your time. If you're looking for a free trial of our SameSpots software, check out the links in the description below. And please feel free to leave any comments, anything you'd like to see in the world of 2D proteomics, or any feedback you've got about the software, please just let me know.